you reached out to us with a really fascinating request.
Yeah.
You wanted us to look into one of the most stubborn, decades-old paradoxes in virology.
Yeah, it's a mystery that has driven scientists crazy for years, honestly.
Right. So to start this deep dive, I want you to just imagine your immune system's resting T cell as like a heavily fortified medieval castle.
I like that analogy.
Thanks. Now, picture cell-free HIV, right?
Yeah.
Just a single free-floating virus wandering alone in the fluid of your body, and it's trying to break in.
And it actually gets pretty far on its own.
Exactly. This solitary invader can breach the outer wall, which is the cell's plasma membrane. It drops down into the courtyard, so the cytoplasm. But then it hits a massive wall.
A literal vault door.
Yeah, it gets completely stuck at the keep's fortified vault door, which is a structure called the nuclear pore complex.
Or the NTC.
Right, the NPC. It simply cannot get past this vault to reach the DNA inside. But, and here's the paradox, when that exact same resting T cell is sitting inside a real human lymph node surrounded by other cells, the virus somehow gets the drawbridge lowered and it just waltzes right in.
It's an incredible biological mystery. I mean, for years, virologists observed this binary behavior. In a lab dish, a resting T cell is practically bulletproof against free-floating HIV. The virus just bounces off or gets stuck in the cytoplasm.
Yes, it just sits there.
Right. But inside the human body, HIV easily and, aggressively infects these exact same sleeping cells.
And for a long time, the assumption was just that we weren't growing the cells right in the lab.
Right.
Or that resting cells just inherently lacked the machinery to be infected.
Exactly. Virologists thought, well, if you want to infect them in a lab dish, you have to artificially wake them up by blasting them with chemicals.
But the sources you shared with us today, specifically this really groundbreaking May 2026 paper from Nature, it completely turns that assumption upside down. Our mission today is to figure out exactly how HIV kicks this biological lock without ever waking up the cell.
And the paper, it fundamentally rewrites what we know about viral entry. It proves that the virus itself has evolved this incredibly elegant way to trick the resting cell into opening its own vault doors.
Which is wild. So let's set the scene for the listeners, because the environment inside a human lymph node is very, very different from a flat plastic petri dish.
Oh, yeah. Completely different. In your lymphoid tissues, cells aren't just, floating around in isolation.
Right.
They are packed tightly together. They're constantly bumping into and pressing against one another. And HIV actually takes advantage of this dense packing through a mechanism called cell-cell spread.
Cell-cell spread. Okay.
Yeah, or CCS. So when an already infected immune cell presses right up against an uninfected target cell, they form this very tight, sustained physical embrace. It's called a virological synapse.
A virological synapse. So they're basically shaking hands.
More like locking arms, really.
Okay, locking arms.
Yeah, the viral protein on the surface of the infected cell, which is called ENVG, it reaches out and firmly binds to the CD4 receptor on the surface of the uninfected target cell.
But wait, if they're just touching, how does that explain the massive increase in infection? Because I always assumed, and I think a lot of early virologists assumed this too, that cell spread was just deadlier because it was a numbers game.
Right, the brute force theory?
Yeah, like you have a giant infected cell pressing against a target, so it just dumps a massive payload of virus right onto it. High multiplicity of infection, I think it's called.
Exactly, high MOI. That was the prevailing theory for a long time. The idea was just if you throw enough spaghetti at the wall, eventually something sticks.
No.
But the researchers behind this new paper, they suspected there was much more to it than just overwhelming numbers. They had a hunch that the physical touch itself, that sustained connection between the N protein and the CD4 receptor, was secretly doing something to the target cell.
But how do you even prove that? How do you separate the physical touch of the infected cell from the actual delivery of the virus?
Well, because if they're touching, the virus is crossing over. Right.
Yeah, exactly.
So that is the genius of their experimental design. They engineered a mutant version of HIV called NVF522Y.
Okay, NVF522Y.
What does that do?
What this mutation does is it selectively breaks the virus's fusion machinery. So the fusion peptide is totally non-functional.
Oh, wow. Let me make sure I'm wrapping my head around this. They build a virus that can still form that virological synapse-like. It can still lock arms and bind to the CD4 receptor, but it physically cannot enter the cell.
Exactly. It's like having the right key to fit into the lock, but the key is physically incapable of turning.
That is so cool. Right. The mutant virus binds to the target cell. It creates the physical handshake, but its membrane cannot fuse. It is permanently locked outside the cell.
That is incredibly clever. So what actually happened when they tested this mutant?
They set up a scenario they called VS priming. They took resting T cells and exposed them to infected cells carrying this mutant virus.
Okay.
So the resting cell gets the firm handshake, but absolutely no virus enters its cytoplasm. None.
Right.
Then they waited. 24 hours later, they took those same resting cells and challenged them with normal, fully functional, free-floating HIV.
And the results were just mind-blowing.
Yeah.
The target cells that got that mutant handshake where, again, no virus actually got inside, they were massively infected by the normal free-floating virus later.
Basically massively infected.
But the resting cells that never got the handshake, they remained completely resistant. The touch itself, just the touch, changed the biology of the cell.
And the researchers actually confirmed this by looking at the surface of the resting cells. The target T cells started pulling their CD4 receptors inside.
Oh, like downregulating them.
Exactly, downregulating them. This is a classic hallmark of a productive, deep HIV infection. It proved the virus wasn't just, stuck in the courtyard anymore. It was actively replicating in the keep.
And crucially, and I want to make sure I stress this, the resting cells never became fully activated, right? They stayed biologically asleep, but their defenses had been completely disabled.
Exactly. They were still resting cells.
It's like a heist movie. You have an accomplice walk up to the front gate of the castle and distract the guard with a friendly conversation.
Right.
And somehow, just by engaging the guard in conversation, the internal alarm system of the entire castle is quietly disabled.
That's a great way to put it.
So an hour later, the main burglar could just walk right up to the back door and slip inside unnoticed.
It's a perfect analogy. It proves that cell-cell spread isn't a brute force numbers game. It is a highly sophisticated, calculated signaling event that alters the target cell's internal defenses.
Okay, so taking a step back for you listening at home, let's make sure we have this foundation locked in. This is our first mini recap.
Sounds good.
We've got a virus that is completely shut out of resting cells when it's floating alone. But when it's delivered via cell-to-cell contact, that physical handshake primes the resting cell, making it incredibly vulnerable to infection. And they proved this using a mutant virus that can only shake hands but can't enter.
Right.
So my question for you, the listener, is this. Think about the environments in your own body. Why would a virus evolve to wait for a crowded room rather than acting alone in the open fluid?
It's a great question to keep in mind, which naturally leads to the next massive question the researchers had to answer.
Right. If the virus is just leaning on the doorbell on the outside of the cell, what exactly is happening on the inside?
Exactly. Where is the traffic jam and how does the doorbell clear it?
Because initially, if you are a virologist looking at this, you might think the bottleneck is reverse transcription.
Yeah, that's a very common assumption.
That's the famous step where HIV turns its RNA into DNA once it's inside the cell, right?
You might think that's where the block is, but the data pointed in a completely different direction. When the researchers measured the viral DNA levels, they found that reverse transcription was actually happening just fine.
Even in the sleeping cells.
Even in the non-primed resting cells. The virus was making its DNA, but it was just piling up in the cytoplasm. The real bottleneck, the true impenetrable wall, is a process called nuclear import.
Ah, nuclear import. So getting into the nucleus.
Right. The massive protein shell of the virus, the capsid core, was getting physically trapped just outside the cell's nucleus.
And the way the researchers visualize this is, straight out of science fiction. They use super-resolution microscopy.
Yeah, specifically technologies called ISIM and DStorm.
Right. I don't want to get bogged down in the acronyms, but basically instant structured illumination microscopy, right?
Yeah.
And DStorm. They engineer the HIV capsids to literally glow bright green.
Yeah, using GFPCA, so glowing green capsids.
So they could look through these incredibly advanced microscopes and literally watch the break-in happen in real time. What did it actually look like?
The visual evidence is just stunning. In the unprimed resting cells, the ones that never got the handshake, the researchers saw that 86% of these glowing green capsids were totally trapped.
86%.
Yeah. They were clustered right at the edge of the nucleus, stuck against the structural envelope called the lamin AC boundary.
So just a sea of green dots backed up against a dark wall.
Exactly. Only a minuscule fraction ever made it inside. But then, when they looked at the cells that had received the VS priming handshake,
the traffic jam just vanished.
it completely cleared up. In the prime cells, 31% of the capsids were actively engaging and moving through the nuclear envelope, and 12% were fully inside the nucleus within just 6 hours.
Wow. It was night and day. So this proves the physical gate is opening. But let's talk about that gate, the nuclear pore complex, because we tend to think of pores like holes in a sponge, right? Just empty space that things flow through.
Oh, but that's not what this is at all.
Right. Explain that to us.
The nuclear pore complex, or NPC, is a massive, highly complex macromolecular machine. It's made of over 30 different types of proteins, which are collectively called nucleoporins, or NUPs.
NUPs, N-U-P-S.
Yeah, and it has distinct parts. It has a cytosolic ring facing the outer cell, a central channel that's filled with this dense, gel-like mesh of proteins, and a structured basket extending into the nucleus.
Okay, so it's a highly regulated, active security checkpoint.
Very active. The virus isn't just squeezing its way through a tight hole. The pore has to actively remodel its shape to let something as massive as the HIV capsid through.
And the researchers noticed a really fascinating side effect of this, right?
They did. After the target cell received that CD4 handshake, it wasn't just the virus that got in faster. Normal cellular transport proteins.
Like things the cell uses for its own daily maintenance.
Exactly. Things like a molecule called important beta-1 or KPN beta-1. They started rushing into the nucleus at a much higher rate, too.
Okay, I want to use an image memory tool here for our listeners to really picture this. Visualizing the nuclear pore is like looking at one of those heavy, automated sliding doors at a grocery store.
Oh, I like this.
Normally, for a resting immune cell, the motion sensor on the door is completely disabled. The door is locked tight. But that virological synapse handshake on the outside of the cell, it acts like someone flipping the breaker box. The motion sensor turns on.
Right.
Suddenly, the heavy doors slide wide open, not just for the viral burglar trying to sneak in, but also for all the normal cellular groceries that the cell is trying to bring inside.
That's a brilliant analogy because it really emphasizes that the nuclear pore complex acts as an active dynamic sensor. It actively responds to signals from the outside world.
So just to recap where we are on this journey, this is our second milestone at the 10-minute mark.
Let's do it.
The primary barrier saving your resting T cells isn't stopping the virus from changing its RNA to DNA. The real barrier is the nuclear pore complex. And by tracking glowing green viruses, scientists proved that the cell-to-cell handshake physically forces this pore to dilate and remodel, letting the massive viral payload slip through.
Perfectly summarized.
So here's my question for you listening.
Yeah.
If you could see inside your own cells right now, you'd see thousands of these gates opening and closing. What does it tell you about viruses that they don't just break the door, but trick the cell into opening it for them?
It really shows how evolved they are.
It does. But honestly, as I'm thinking about this, I'm losing the thread on one major logical point. I have to push back here.
Go ahead. What's the missing link?
If touching the CD4 receptor on the outside of the cell is the master switch that opens the nuclear door, why wouldn't a single free-floating HIV virus just bump into that exact same CD4 receptor, touch it, and trigger the exact same alarm system?
Right.
Like, why does it only work when a whole infected cell does the touching?
It is a glaring physical contradiction, and it's exactly what the researchers had to solve next. And to do it, they designed what I think is the most elegant part of the entire study. They called it the bead experiment.
The bead experiment.
Yeah, it all comes down to physics and scale.
Physics. Okay, how did they use beads to solve a virology problem?
So they took artificial synthetic beads and coated them with either HIV-NV proteins or specific CD4 antibodies. But the real trick was they used two completely different sizes of beads.
Okay, size matters here.
Hugely. First, they used tiny beads that were exactly 130 nanometers in diameter.
Which is, wait, that's the precise physical size of a single free-floating HIV virion, isn't it?
Exactly. So they basically made little plastic clones of the free-floating virus.
Got it. And when they sprinkled these tiny 130 nanometer beads onto the resting T cells?
Absolutely nothing happened. The nuclear pore stayed shut tight. The infection rates stayed exactly where they were.
But then they brought out the heavy machinery.
Yes, they did. They used giant 5-micrometer beads.
5 micrometers. To put that in perspective for the listeners, 5 micrometers is roughly the size of an entire T cell.
It's massive on a cellular scale. So when they coated these massive beads with the viral proteins and applied them to the resting cells, it perfectly mimicked the cell-to-cell spread.
Oh, wow.
The nuclear pore unlocked, and the resting cells became massively susceptible to infection.
I love this because it makes the microscopic world feel so tactile,
Yeah.
This process, it's called macro cross-linking, right?
That's the scientific term, yeah. Macro cross-linking.
It's like, imagine you have a very heavy, stiff mechanical security button on a wall. If you take a single grain of sand, which represents our tiny free-floating virus, and you press it against the button, you simply don't have enough surface area or physical leverage.
Right, the button won't push in.
Exactly. But if you take your entire thumb, the giant 5-micron bead, or a whole infected cell, and press that same button, you easily trigger the mechanism.
That is exactly what receptor clustering is. A single 130 nanometer virus is physically too small to pull enough CD4 receptors together on the surface of the target cell.
Right.
But when you have the massive flat surface area of a whole cell pressing against the target, it physically grabs and groups dozens or even hundreds of these CD4 receptors together in one spot.
And that massive grouping is the physical leverage required to trip the internal cellular alarm system.
Precisely. Scale is everything in cellular mechanics.
Okay, mini recap number 3 for everyone listening. Free-floating HIV is simply too small, 130 nanometers, to trigger the required signaling. It requires the massive surface area of a whole infected cell 5 micrometers pressing against a target to cross-link enough CD4 receptors and trigger the unlocking mechanism.
Spot on.
So my question for you, think about scale in biology. How often do we assume a chemical reaction works just based on the ingredients, right?
Yeah.
Without considering the physical size and leverage required to actually make it happen.
It's a completely different way of looking at biochemistry.
It really is.
Yeah.
But now that we understand how the giant thumb presses the button on the outside of the cell, we have to trace the electrical wiring inside.
Yes, the signaling pathway.
How does a signal travel all the way from that cluster of CD4 receptors on the plasma membrane deep through the cytoplasm to actually reach and physically alter the nuclear pore complex? What is the molecular domino effect here?
Well, the researchers meticulously mapped this out step by step, and it relies on what we call a kinase cascade.
Okay, let's define that. What is a kinase cascade?
A kinase is simply an enzyme that modifies other proteins by chemically attaching a phosphate group to them. You can think of it like passing a baton in a relay race or flipping a series of molecular light switches.
I like the relay race idea. So who is the first runner?
Step 1. When those CD4 receptors cluster together on the surface, they immediately activate an internal kinase called LCK.
LCK. Let me stop you there, though. How do we know LCK is the first domino? Like, how did they prove that?
Good question. Because the researchers applied a specific chemical inhibitor that blocked LCK from working. Okay. And when they did that, the entire process halted. The nuclear pore stayed completely shut, and infection dropped back to 0. The domino effect was killed at the very first step.
Okay, so the surface receptors group together, they flip the switch on LCK. What does LCK do next in the relay race?
LCK then triggers a cascade of downstream proteins. You might see them in the paper as ZP70, ERK, or AKT.
ZP70, ERK, and AKT. That's a lot of alphabet soup.
It is, and we don't need to get too lost in every single middleman. The most important thing for us to understand is the final destination of this signal. The ultimate target of this entire cascade is an enzyme called CDK1.
CDK1. Now, this is where my jaw genuinely dropped when I read the sources.
I know exactly why.
Because anyone who remembers their high school or college biology might recognize CDK1. It is famous for being the master controller of cell division, right? Mitosis.
Yes, exactly.
It's the engine that tells a cell to split into two.
Wait, you said earlier that the resting T cell doesn't wake up? It isn't dividing here. And that is the true elegance of this viral hijack. The researchers rigorously tested this. They checked for markers of cell division like Ki67 expression, and there was absolutely no DNA synthesis happening.
Wow.
The cell was not dividing. What HIV is doing is surgically hot-wiring just a tiny sliver of the cell cycle machinery to manipulate the nuclear pore without ever turning on the whole car.
That is terrifyingly efficient. So how exactly does CDK1 unlock the door?
Well, to turn CDK1 on, the cell has to do some very specific chemical editing. It's actually really complex. The cell has to remove inhibitory phosphates from certain spots on the protein, specifically amino acids 3,14 and tar-15.
So that's like taking the parking brake off.
Exactly, taking the parking brake off. And then it has to add an activating phosphate to another spot called Thur161.
Which is hitting the gas.
Yes. It also blocks a protein called WE1, which normally inhibits CDK1. So once CDK1 is fully activated, it acts as the master locksmith. locksmith.
And how did they prove this locksmith activity? Because looking at proteins is hard.
They proved it using mass spectrometry or phosphoproteomics.
Right, which is essentially a highly sensitive molecular scanner that can read the exact chemical tags on proteins. Right?
Exactly. It tells you exactly what proteins have been phosphorylated.
And what did the scanner find at the nuclear vault door?
They saw that activated CDK1 was directly phosphorylating, meaning physically attaching chemical tags to specific nucleoporins right at the gate.
The NUPs we talked about earlier.
Right. Specifically, it targeted NUP54 and NUP62.
And where are those located?
Those are the proteins that make up the dense, gel-like mesh inside the central channel of the pore.
Oh, okay.
It also tagged a protein called TPR, which is part of the inner basket facing the nucleus.
So by chemically tagging these specific mesh proteins, CDK1 causes the entire structural matrix of the pore to relax, dilate, and remodel.
Precisely. The chemical tags change the physical shape of the proteins, granting the massive HIV capsid the clearance it needs to finally slide through and deliver its DNA.
Let's trace this entire wire from start to finish for our listeners, because this is our final mini recap, and it's the core of the whole discovery.
Let's trace it.
The journey starts when a massive infected cell presses against a resting T cell, grouping enough CD4 receptors together to act as a giant physical key.
Sure.
This massive grouping triggers an internal wire, activating the LCK enzyme. The signal cascades down the line through ZAP70 and AKT until it hits CDK1.
A protein normally used for cell division.
Exactly. But here, CDK1 acts as a molecular locksmith, chemically altering NUP54, NUP62, and TPR inside the nuclear pore, forcing the gate to slide wide open so the virus can slip inside.
Beautifully summarized.
So for you listening, think about this. We always hear about viruses mutating to evade our immune systems, right? But here, the virus is using our own perfectly functional proteins, MCK and CDK1, against us. Does this change how you view viral infections?
It really is a complete paradigm shift in our understanding of viral pathogenesis.
Why is it a paradigm shift?
Because for decades, virologists treated the nuclear pore complex as just a passive hole in the wall, and they treated cell-to-cell spread as just more virus. Now we know that the nuclear pore complex is an active, dynamic cofactor for infection.
So the resting cell isn't actually immune.
No, it just requires a physiological cue, that macro cross-linking, that cell-free viruses are physically too small to provide.
It's a hostile takeover of the cellular switchboard.
It really is. And understanding that switchboard opens up entirely new avenues for treatment.
Like new drugs.
Yes, because if we know the exact chemical pathway the virus uses to open the door, the CD4 to LCK to CDK1 pathway, we can potentially design drugs to jam that specific signal.
So locking the vault permanently, regardless of how much virus is outside.
Exactly. We cut the wire before the signal reaches the door.
Which brings us to a final provocative thought for you to mull over as we wrap up this deep dive.
I love this part.
If HIV has evolved over millions of years to hijack the cell's own communication systems, just to politely ask the nuclear door to open, what other viruses might be doing the exact same thing?
That is the million-dollar question.
Right. Think about pathogens like hepatitis B
Yeah.
or the herpes virus. They also have these really large, bulky capsids that have to somehow navigate this exact same nuclear pore.
And they are notoriously known to use cell-to-cell spread inside the human body, too.
Exactly. Could this CD4, LCK, CDK1 pathway be a universal Achilles heel for multiple viruses? Are we on the verge of discovering a single biological lock that we could bolt shut to stop an entire class of diseases?
It's a profound question, and honestly, it's one that virologists around the world are undoubtedly rushing to answer right now. We may be looking at a deeply conserved, universal mechanism of viral entry.
And to think, it all starts with realizing that the castle was never completely impenetrable. You just had to know exactly which brick to push on the outside wall to lower the drawbridge. Thank you so much for joining us on this incredibly dense, rewarding exploration of the microscopic world. We hope this changes how you view the microscopic battles happening inside you right now. Keep questioning those hidden mechanisms, and we'll catch you on the next deep dive.